Hello everyone. This is Angeline Kirbai, Assistant Professor, Department of Biotechnology, St.Josephs College of Arts and Science for Women, Hosur. Today we are going to see about culture media for microorganisms. What is culture media? Culture media is a gel or liquid that contains nutrients and is used to grow bacteria or microorganisms. They are also termed growth media also. Different cell types are grown in various types of medium. Nutrient broths and agar plates are the most typical growth media for microorganisms. Some microorganisms or bacteria need special media for their growth. Significance: culture media is used in order to identify the causative agent from infected material. Types of culture media: the culture media are classified in many different ways based on physical state, based on presence or absence of oxygen, and based on nutritional factors. Based on the physical state, it is classified into two types: liquid media and solid media, semi-solid media. Based on the presence or absence of oxygen, it's further divided into anaerobic media and aerobic media. Based on nutritional factor: simple media, synthetic media, complex media, and special media. First one is common form of media. Nutrient broths or lysogency broth medium are the two most popular growth media for bacteria. Agar is frequently added to liquid media before being placed in a petri dish to solidify. These agar plates offer a stable medium for the cultivation of bacteria. It stays solid because relatively few bacteria can break down agar. Classification based on nutritional component. Simple media. It is a general purpose media that supports the growth of non-fastidase microbes, and it is primarily used for the isolation of microorganisms. Example are nutrient broth, peptone water, and nutrient agar. Complex media. These are media containing nutrients in unknown quantities that are added to bring about a particular characteristics of microbial strain. Example are the tryptic soy broth, blood agar, and nutrient broth. Classification based on the nutritional component. That is synthetic media. Synthetic media is a type of chemically defined media and is produced from pure chemical substances. A defined media refers to a medium having a known concentration of ingredients like sugar and nutrient sources. It is generally used in scientific research. An example of this is Swebek dogs medium. That is then next one is special media. Special media are seven different types. Basal media. These are routinely used simple media having carbon and nitrogen sources that boost the growth of many microorganisms. They are also known as general purpose media and are considered non-selective media. Enriched media. This media is prepared by adding additional substances like blood, serum, or egg yolk in the basal medium. It's used to grow fastidase microorganisms as they require additional nutrients and growth promoting substances. Example of enriched media are chocolate agar, blood agar, and loeffler's serum slope. Selective media. This media allows the growth of certain microbes while inhibiting the growth of others. It's an agar based medium that is used to isolate microorganisms in labs. The selective growth microorganisms is decided by adding substances like antibiotics, dyes, bile salts, or by pH adjustments. The next one is differential media. It's a term used to describe a medium that has components that aid in identifying the various properties of bacteria. Peptone, agar, lactose, neutral red, and sodium taro chocolate are all ingredients in Mekanki medium. Here, the colonies made by lactose fermentation are pink, but those made by non-lactose fermentation are pale or colorless. Next one is transport media. The special media developed for the transportation of bacteria to laboratories from a certain distant places of sample collection, and these are known as transport media. Examples: toward transport medium. Indicator media, next one. When the bacteria multiply in these media containing an indicator, they tend to change their color. Mekong medium is also an example of indicator medium. Another example is the black colonies of Salmonella typhi that develop on sulfate containing Wilson and Blair media. Sugar media. It contains 1% sugar, which can be any fermentable substances like glucose, mannitol, sucrose, and lactose. The generation of acid following the fermentation of sugar transforms the medium into pink due to the presence of an indicator and also show gas formation. Conclusion. Culture media or growth media is a liquid or gel-like substances that provide essential nutrients and minerals required for organisms to grow under laboratory conditions. All microorganisms have different requirements based on their habit and habitat. Thus, culture media needs to be formulated in such a way that they support the organism's life in a lab. Thus, a variety of culture media have been developed by scientists according to the type of organisms they want to study and the purpose of their experiments. Thank you.